Good morning, class. You're welcome to Biotechnology and Animal Production AMP 504. Last week, we looked at the basic concept of biotechnology, recombinant DNA, and genetic engineering. However, this week we'll be looking at the tools for those basic concepts. I know you are going to have an exciting time as we go to the class. I am Dr. Awolumate Samuel, your facilitator. Welcome class to Biotechnology and Animal Production. We are in the second topic, genetic engineering tools. I remain your facilitator, Dr. Olumate Samuel. Last week we were looking at GMO food, products of biotechnology, recombinant DNA, and genetic engineering. We also look at test tube baby. We look at the different animals that were products of genetic engineering and recombinant DNA. This week, we will want to ask some basic questions. Have you ever seen an identical twin from two mothers? Is that possible? Yes, it is. That is the basis for this course. When scientists make a genetic copy of a sheep, this is an example of a clone. All the picture you see here are example of a clone. And so what is a clone? An organism or cell that has the same genetic makeup as another, the same genetic makeup as another. Okay, and so that's the tools that we use in making them is what we want to bring to your attention. Okay, so DNA is a basic recipe for our body. We'll soon define it accurately. And so what are the steps for making those DNA food that we saw previously? What are the various steps? This will be our learning outcome in this class. And finally, we'll be describing to you what we call marker-assisted selection and its usefulness. So that means, what do you think about their importance? You will see their importance as the class progresses. Of course, you may say, I hear you say, I don't even know all of these things. Well, that's why I am here. Okay. So the definition of DNA is a large biomolecule that contains the complete genetic information of an organism. All the genetic information is contained in our DNA. What is the significance? Well, every cell in a living organism has, for example, the virus contains DNA. The basic building block of DNA molecules are nucleotides, and a single strand of DNA may contain billions of these nucleotides, billions of them. Of course, like I said, that is the genetic makeup of our body. So, how is this DNA used in genetic engineering? Well, we have to manipulate this gene. It has to be manipulated. So when we manipulate this gene, we are able to come up with that clone picture that we saw previously. Okay, and of course, what is cloning? We have defined it briefly before. A process of creating a group of individual cells or organisms that descend from one progenitor. So when we have such organisms, that's individual or group of individuals that descend from one progenitor, that's the process is called cloning. So members of a clone are genetically identical because cells replicate their own daughter cell each time this cloning takes place. So what are the examples? These are the examples we showed you before. These are examples of clone human being, sister clone dolly sheep. Of course, that's the dolly sheep from which these four sister clones were cloned from. And what is the process? Now look at the process as we explained. From unfertilized egg, as you can see in this picture, we move to the nucleus was removed. The cell, each of this egg, at this point have no nucleus. Then from this organism, which we want to use for cloning, we remove the cell from the body, and the cell that is removed from this body is now used, is not manipulated with this egg here, and we begin to produce embryo. And that embryo, is implanted in a different sheep, and we produce chip of this same type before. So that is the process of cloning. Okay, so what is the usefulness of this cloning? Number one, to amplify DNA fragments containing whole gene, to also expand DNA sequence, such as the promoters, the non-coding sequence, and random fragments. And finally, a wide range of biological experiments and practical application, like genetic fingerprinting, protein production, and that's what we use this cloning to achieve. The essential step, involved in cloning DNA fragments. What are they? Fragmentation, litigation, transference, screening, and selection in that sequence. So fragmentation just means the breaking apart of the various strands of this DNA. And litigation just means the gluing together of these pieces of DNA that we have, broken into different parts, into fragments before. And we now insert this new form of piece of DNA into the cell, like the picture we showed you earlier. And after that, we screen out the cell that were successfully transferred into another new DNA. What is the technique of this genetic engineering? Number one, we identified an organism that contained undesirable traits. Then we extract the entire DNA from the organism. The third step is remove this gene from the rest of the DNA. Finally, we have to insert this DNA, this gene, into an existing, organism DNA. Next, we are going to look at what is marker-assisted selection. What is this marker-assisted selection? We want to look at it closely. It's an indirect process where a trait of interest is selected based on, some certain characteristics, the morphology, the biochemical component, the DNA, variation. And this selected trait of interest is now introduced into another trait of interest again that has, for example, it could be productivity, disease resistance, abiotic stress, tolerance, and quality. So when we do all of that, we are assisting the selection process because we have looked at, traits that are of interest to us, and we connect it to another trait of interest. Rather than just looking at the trait itself, we look at, the genetic component of it, and that is marker-assisted selection. Commonly used molecular marker. What are they? Number one, we look at amplified, fragment length polymorphism. We have restricted fragment length morphism, random amplified morphism, simple sequence repeats, or microsatellites. Then finally, we have the single nucleotide polymorphism. These are the common markers that we use in selecting all of those traits. And what are the 5 important steps for this marker aid selection? What are they? Number one, select the parents. Parents is very important because the genetic component of that parent, the significance of it, will show up in the final product. So we develop the breeding population. Number one, this DNA is isolated from each plant or animal. And then we score, remember we said we use the molecular marker. We score it according to restricted fragment length morphism. Then finally, we use correlation with those traits that we have isolated. This morphological trait of isolated, we correlate it. If it rhymes together, then we can be sure that the steps that we follow is accurate. Then, what is the importance of this marker-assisted selection? We have 7 of them. It helps us to transfer the maize sterile and also is insensitive into a cultivated genotype from different source. It's a very effective, efficient, and rapid method of transferring resistance to biotic and abiotic stress. We also, look at it in the sense that it's effective in introjection of desirable traits gene from wild into cultivated genotype. If you have a wild gene, and yet it is effective, we can transport it to cultivated genotype. And it's also effective in genetic improvement of plants and animal. So and I didn't mention the second one, genes pyramiding for disease and insect resistance, meaning that when we use marker assisted selection, we're able to have, genes that are resistant to diseases and insects. Okay? Of course, the character of the new crop or animal is also improved. We're able to transfer desirable gene from one cultivar to another, meaning that from one generation of species to another. Now, this picture is showing you that by the time we use marker assisted selection, the cloned animal that we now have for production in mass is heat resistance, the body temperature is able to accommodate 40 degrees centigrade. At the same time, the milk production is very high, the fertilization is accurate and of maximum productivity, the dry matter intake is very high, and you can have maximum productivity. The efficiency of that animal is high because the selection process is also of high quality. In summary, we told you that DNA contains genetic information to determine the organism trait based on the characteristics that we actually needed. And by modifying the DNA, engineers are able to determine which traits of an organism that we want to actually use. What does it possess? Do we need to use it to increase our productivity? Remember, the objective of animal production is sustainability and productivity. To have it in quick succession, disease resistance, its productivity is high. So we also learned in this class that a clone is genetically identical because cell replicate itself, and we now have identical daughter cell each time we produce that's the next generation. We also told you that trait of interest can be selected based on morphology, based on the biochemical, based on the DNA variation. And once we link them to the trait of interest, we're able to have, productivity is high, there's disease resistance, abiotic stress is contained, and we have high quality products. These are what the tools of genetic engineering is able to achieve. And like we said, we use marker assisted selection process to achieve all of those things that we mentioned. Wow. Thank you for listening. I know that it's been an exciting time. I hope you learn new things. I have learned new things. So in the next class, we'll be discussing transgenesis of genetic material. How do we achieve all of those transgenesis, this genetic material? How do we transmit it from one species to another? Now, before I bid you goodbye, I would like to take a short test so that they can be useful reminder of the lesson. What are they? Just write down in your own word 5 vital importance application of marker assisted selection. Try and put together list 5 relevant steps for this marker aid selection process. And you can choose to demonstrate for us the process of cloning with the aid of a well-labeled diagram, like I showed you before. You can also help me provide a clear understanding. What's your clear definition of all of those terms, the cloning, the DNA? Try and define them. Then could you please write down a potential use for the results of cloned animal? What is the results? So write them down as a very good reminder. If you can do all of that, you can attempt the test that will be coming up on Friday. So enjoy the reading of the notes, attempt the class on Friday. Hope to see you next week. Bye-bye.